Hello, my name is Natalie Rico, and I'm doing a neuroscience poster. Um, my poster is on the role of hunchback in regulating Drosophila brain development and behavior. This is in collaboration with Dr. Kristen Lee and Dr. Christo. So a little background on my poster itself and what I work with. So I work with neurons, and they're cells that communicate with each other through signals. So neurons have dendrites that receive signals from other neurons, the cell body that decides whether or not these signals are important enough to go to communicate with other neurons, and then the axon and axon terminal that communicate with those other neurons. And so I work with Drosophila, which are fruit flies, because they have very similar neuronal circuits as us, and they are just very similar to humans, both cellularly and molecularly. And so, like all organisms, they have something called transcription factors, which are proteins that turn genes on and off by binding to DNA sequences. And this is a form of gene regulation that all organisms have. And so within the Drosophila system, I'm using the well-characterized moonwalking descending neuron as its transcription factors are known, and its behavioral output of backwards motion is observed in both the larvae and the adult. And so in the moonwalking descending neuron, uh, there is a cell body, which you can see here. There's a cell body, the ipsilateral dendrites, which are just on the cell body side, and then the contralateral dendrites, which are on the opposite side, and then as well as the axon. So they have all the morphological uh, uh, landmarks as all neurons do. And so I wanted to see how MDN works within the Drosophila. And this is very crucial because understanding how the regulation of neurons affect behavioral output and morphology can help further understand motor neurons in general and also motor disorders, how they work, and how they could possibly be treated in humans later on. So, previous work has shown that MDN expresses the transcription factor hunchback in the Drosophila larvae and the adult, and that the moonwalking descending neuron has two neuronal partners, pair 1, which is associated with pausing, and A18B that has the unknown role, as you can see here. Uh, pair 1 is in the brain, and A18B is in the VNC, which is the ventral nerve cord, and this is analogous to a human spinal cord. And then this just shows that hunchback is expressed in MDN. So I wanted to see how hunchback is affecting MDN within the Drosophila larval brain. So to do this, I wanted to take away or knock down hunchback. So I used a tool called RNA interference, also called RNAi, to inhibit gene expression, also called gene knockdown. And so we can see this here, how it essentially binds to the specific RNA that was going to make that protein, for example, hunchback, like I'm working with here, and essentially dices up that RNA so it won't be able to make hunchback. And then so the three pillars of data that I wanted to look at specifically are behavior, morphology, and partner analysis. So if anything's happening between MDN and its neuronal partners. So for the behavioral analysis, I used a tool called optogenetics, where a red light is shown onto the larvae, and that activates MDN. This is done by using the Gal4 binary system to express crimson, which is a transgene that is gives MDN the ability to be activated using red light, and then we captured the behavior on a video. And then for the neuronal partner analysis, I used immunohistochemistry. So we dissected larval brains and then stained for MDN and its neuronal partners that we were interested in. So that's that. And then for the morphology, we use something called MCFO, which is also called multicolor flip out, which is a technique to label individual neurons, as you can see here, or one or two neurons. This again uses the Gal4 binary system to target specific tissues, in this case MDN. And also there is another level of control using heat shocking to remove a stop codon in the UAS promoter system. So that is why we are able to only label one or two neurons because MDN in general, without so regular labeling of MDN, these are actually like four neurons. It could be actually eight, but there is no distinction between the dendrites as you can see here. So imagine like four of these overlapping each other. We can't really see if there's any morphological changes happening. So MCFO gives us that ability to see it. And so the big overall arching idea was that Hunchback, of course, is a transcription factor, so it's affecting MDN, and we know that it's getting expressed in MDN. So my hypothesis was that manipulating the transcription factor Hunchback in MDN or the moonwalking descending neuron will affect its morphology, its behavioral output, and its relationship to its neuronal partners. So to get into the data, I first started out with behavior because we knew that there was a behavioral output. So I wanted to see what would Hunchback do when it's knocked down to the behavior. So um, we saw that when Hunchback was knocked down, the larvae actually went backwards farther and they went backwards faster and for a longer time frame. So this shows the many larvae data points that we had, and you can see here that the Hunchback larvae significantly has significantly increased di backwards distance than does the control. And then here we see that during this red light therapy, where MDN is getting activated using optogenetics, during this time, they're actually going backwards faster. So that means that the speed is going in the x-axis, so it's going down more. And also it is going backwards for a longer time. So this made me think that something is changing in MDN when Hunchback is knocked down. What could that be? So my first guess was that some sort of anatomical or morphological changes are happening that is affecting MDN's regular behavior. So that is the next um, kind of data that I looked into. So I wanted to know, does Hunchback regulate MDN's morphology? So when we did the same thing using RNAi and RNAi knockdown with Hunchback, and we used the three anatomical landmarks that I discussed earlier. So the ipsilateral, contralateral, and dendrites and the axon. And so what we saw, what we were looking for specifically is branch number, and this just means how complicated or bushy the dendrites and axons are. And then we also want to see summed length, which is essentially just how long it is. And so for both the ipsilateral and contralateral dendrites, we saw a significant increase from control in branch number, so how bushy and complicated they were, and also a significant increase in the axon branch number. In addition, we saw a significant increase when Hunchback is knocked down in the summed length of all three of these anatomical landmarks, the ipsilateral dendrites, the contralateral dendrites, and the axon. And so this led us to think that the MDN has its neuronal partners, and this is in a descending fashion. So MDN's up here and it's connecting to these, usually through the axon. So we wanted to see if the axon itself is connecting to new neuronal partners or possibly connecting to A18B, which has an unknown role. So our next our next kind of experiments were to see if Hunchback prevents further interaction with other neurons like A18B. So we looked specifically at A18B. Here we stained for MDN and A18B. And as you can see, this is the control. And MDN only goes so far as A4. And these are just using an Eve staining, which just stains for the specific abdominal sections of the ventral nerve cord, which again is analogous to our spinal cord. So it only goes around A4. Um, But we know that A18B goes down. It starts at A4 and then it goes down all the way to A7. When Hunchback is knocked down, as you can see here, the MDN axon actually goes below A6, the abdominal section A6. So this led us to think that MDN and A18B might be interacting, or some sort of communication might be happening between MDN and A18B that could be an explanation for this behavioral phenotype. And what we saw was that the number of A18B and MDN overlaps was significantly increased in the hunchback knockdown larvae. This doesn't necessarily mean that they're communicating more, but it does give a really great insight to where my project will go further. So, some conclusions for all of this data is that hunchback is required for the regulation of backwards motion, which is the output of MDN in the larvae. Without hunchback, the larvae's backwards distance has increased speed and distance. In addition, hunchback regulates branching and axon length into the lower abdominal regions, as I shown before, in the larval brain, meaning that hunchback limits the growth and complication of dendrites and the extension of the axon to lower regions where it could be communicating with more or new neuronal partners. Um, Without hunchback, it overlaps with more A18B neurons, one of MDN's neuronal partners. Hunchback could be communicating more synapsing, which are just points of communication between neurons, with A18B, which could explain the behavioral phenotype of increased backwards motion. And that kind of goes into my future directions. So I want to see, are there synaptic connections happening between MDN's neuronal partner, A18B? In addition to the overlap that is clearly shown here. Um, and if so, are these points of communication adding to that behavioral out phenotype that we got of increased backwards motion? I also want to see if maybe MDN is communicating with other neurons or other parts of the abdominal sections of the ventral nerve cord, and whether or not that is affecting those areas, because those areas are in control of different parts of the body, and perhaps MDN is now regulating or controlling new parts of the body. And then also, I would like to see if this mechanism is conserved in the adult flies. What happens when hunchback is knocked down? Does the behavioral output change, or does the morphology change? And whether or not hunchback or whether or not MDN is communicating with A18B. And so that is my poster. Thank you.